All right, thank you for attending today's webinar. I'm going to kick things off by talking about how the new Ampli-Fi protocol enables the sequencing of low input and challenging samples. Now, at PacBio, our latest HiFi sequencing platforms address a set of diverse needs in the genomics community. Vega is our low capex benchtop system, ideal for lab sequencing around, let's say, about 100 genomes per year. Whereas the Revio system is a larger system, a production scale system capable of sequencing thousands of long-read genomes annually. Now, one long-standing challenge when it comes to long-read sequencing has always been the DNA input requirement, right? Especially if you're doing PCR-free workflows. So over the years, we've really focused and made significant progress on reducing this barrier. With our latest Spark chemistry update on Revio, we've brought that DNA input down to just 500 nanograms. But this is a major improvement. DNA availability, continues to be a limiting factor for certain sample types and organisms. So to address this, we've sort of modernized what people have kind of known previously as the ultra-low input workflow, right? And we've moved that over to the Revio and Vega platforms. We're calling this new iteration Ampli-Fi, short for amplification and HiFi sequencing. Now, with Ampli-Fi, we've achieved several key improvements that I want to highlight. The first one being, it represents a 50% reduction in the prep costs for doing single samples compared to the older protocol, right? We've also added support for larger genomes from a single prep. The previous protocol was sort of limited to 500 megabases. With the new protocol, you can do genomes up to 3 gigabases. We've broadened that DNA input range. You can use DNA inputs all the way down to 1 nanogram, upwards to 50 nanograms. We've also reduced the number of PCRs that you need to do. The previous protocol had 2 sets of PCRs that you then had a pool. The new protocol just uses a single PCR. We've also increased the reaction capacity by using SmartBelt Prep Kit 3.0. And each of the preps are going to give you the ability to run multiple SMRT cells. Now, these changes provide our users multiple benefits. The first being just, access to more samples on your Revio and Vega systems, and you can use as little as a single nanogram of DNA. The workflow itself is much more streamlined. It only takes a single day to do. And the data quality is going to be better because you're going to get more uniform coverage and reduced PCR bias. And we see this when comparing the new Ampli-Fi protocol to the older ultra-low DNA input protocol. Ampli-Fi has higher amplified DNA yield, it has lower duplication rates, it has improved coverage, especially in those high GC regions, and just better, more uniform coverage overall, while at the same time maintaining comparable HiFi read lengths to the older protocol. Now, a key part of this improvement comes from switching to the KOD Extreme hot start DNA polymerase from Millipore Sigma. This replaces the previous method that relied on 2 separate enzymes requiring 2 PCR reactions and a pooling step. Now, this improvement in coverage results in a more contiguous and complete human genome assembly. And this was shown in a recent publication from the group at the Loeb Center for Translational Biodiversity Genomics in Germany. So even at 23x coverage, you can get multi-megabase contig N50s with 98% gene completeness. This is more than a 4-fold improvement over the previous protocol. So for example, the contig N50 for HAP1 increased from 642 kilobases using the old protocol to 2.8 megabases using KOD Extreme. So this was also proven in other organisms. So again, the same group in Germany tested KOD Extreme. The tested KOD's extreme performance on assembly performance on various genomes, including the man sloth, and several mollusk and arthropods, really proving out its robustness. Now, moving back to humans for a second here, the Ampli-Fi protocol can also be used for variant detection when there really isn't enough DNA for standard amplification-free workflows, right? So there's strong performance across all the different DNA input levels at 30x coverage, with single nucleotide variant calling performance being the most comparable with standard PCR-free sequencing, while the indel and structural variant performance are more impacted from amplification, which is, to be expected. But the results are still very good, especially considering the benefits you get with the ability to phase when using long reads. Here's another sort of practical example showing how the Ampli-Fi protocol improves coverage. So here in that top panel is data generated using the Ampli-Fi protocol. And you can see here the reads span the entire high GC rich promoter of the FMR1 gene, whereas the bottom panel is data generated by the old ultra-low input protocol. You can see here we have a coverage gap, mainly because the inability of the polymerases used in that protocol to amplify and extend through this high GC rich region. Now, Ampli-Fi features a one-day streamlined workflow. I don't have the time here to go step by step. I just want to highlight some of the things that have changed relative to the old protocol. So one thing we've done is optimize and updated the sharing parameters for the Megaruptor 3 and Covaris G-tube systems. We've also optimized it where you do not have to do a bead cleanup after sharing. We've also switched to using the Twist universal adapter system with to enable dual indexing during the amplification step. We've also moved to using a single polymerase, as I mentioned, the KOD Extreme Hot Start DNA polymerase. We've also moved to switching over to the SMRTbell Prep Kit 3.0 chemistry. This way you can use the same kit for both your PCR and PCR-free workflows. We've also included the option to use bead-based size selection in the protocol, so no longer do you need to do a gel cassette size selection. And finally, we've integrated the polymerase binding step directly into the protocol, so no longer you don't have to go to SmartLink to do your sequencing primer annealing and your sequencing polymerase binding. Because the protocol, uses these unique dual indexing at the amplification step, the samples can be pooled upfront of library prep for additional cost savings when working with small genomes like microbial isolates or metagenomes, for example. At PacBio, we tested the protocol on a microbiome standard for which we had previous amplification-free data for. So we had 2 users do 2 independent preps. One was for the Revio system, one then went to the Vega system. Both preps, both sequencing experiments produced really high quality results with a minimum duplication rate and a high demultiplexing rate. I just want to kind of note that the difference here between that duplication rate on the Revio and Vega system is likely just a result of the stochastic differences between doing just 2 independent PCRs. And it doesn't reflect any kind of systematic difference between the 2 systems. Now, we've done a number of analyses on these data sets. I don't have time to go through all those, but I did want to show you this one particular set of analysis doing metagenome assembly from these data sets. Now, before we could assemble them, we did have to normalize the coverage between the different data sets. So we downsampled to 2 million reads, which was the depth that we had of the standard PCR-free data set. So at that 2 million read coverage, we really didn't notice any difference between the metagenome assemblies for the Ampli-Fi protocol versus doing it with PCR-free. We also didn't notice really any differences with some of the other analyses looking at taxonomic identification. So there was really no red flags or clear biases that the protocol was introducing. So it seems like a really good way to sequence, some of these more DNA limited samples with this new protocol. I will just caveat that, this, I do concede that this is a fairly simple metagenomic sample. So the situation could be fairly different with a more complicated metagenomic sample, but we're going to be doing more testing on that later. In addition to making it easier to sequence metagenomic samples, the protocol enables advanced applications like CiFi, which was developed by the Dennis Lab at UC Davis. Now, CiFi is a way to perform the 3C protocol with HiFi sequencing to get multi-contact chromosomal information, from a single read. The reason why Amplify enables this is because the amplification step eliminates those residual crosslinks from the 3C protocol, allowing for the efficient HiFi sequencing of the reads. Now, Scott Geib, he's going to go in a lot more detail and in depth on the CiFi application. So later in the webinar, so stay tuned on that. Another thing we're doing in the Amplify protocol is really exploring FFPE samples as the next frontier for this workflow. Early tests show that the protocol can work well on some FFPE samples, like in the case shown here. Though the reads, from FFPE samples tend to be, short in the long read sort of, space. We're talking 1 to 2 kb in length. They're still long enough to sort of phase entire genic regions, like shown here for the EGFR gene. And this is important because it's going to help for the detection of compound hets or structural variants in some of these important genic regions. Okay, so I just want to leave you with a few key takeaways on the Amplify protocol. So we've shown that the protocol can reduce the DNA input requirements for Revio and Vega systems. It has a number of workflow and performance improvements. It has a much lower cost and complexity relative to the older workflows. And it's really going to enable access to a lot more samples for long-read sequencing on the Revio and Vega platforms. And it's also going to enable those advanced applications like CiFi and doing long-read sequencing on the FFPE material. So with that, that concludes my section. So Mike and Ksenia from the Wellcome Sanger Institute are up next, and they're going to talk about their experience with the early access Amplify protocol and applying it to the Darwin Tree of Life program.
Hello, everyone. My name is Michael Quail. I'm Principal Scientist for Sequencing R&D at the Wellcome Sanger Institute, and I'm going to talk about some of the work we've done with PacBio, with Ampli-Fi and ultra-low input. So Sanger Institute has a history of de novo sequencing projects. One of our largest programs at Sanger is Tree of Life. The Tree of Life project is doing a lot of projects under the general umbrella term, the Earth Biogenome Project, aimed at sequencing all of eukaryotic life on Earth. One of our largest projects is the Darwin Tree of Life, which is aiming to sequence all life in the British Isles. So as part of this, we've developed the genome engine, where samples are acquired. We extract RNA and DNA. We do genome and transcriptome sequencing, so specifically long read on Pacific Biosciences Revio sequencer, HiC and RNA-seq on a short read platform. We assemble those long reads, scaffold with HiC, and use the RNA-seq data to annotate. So assemblies are done via our assembly group, and you'll hear more from one of our bioinformaticians, Ksenia, later. All our projects and the data is tracked via the TOLQC webpage. Typically, we would aim for 25x HiFi data, which is assembled using HiFiasm. We then remove duplicates, scaffold that assembly from, say, the HiC data using yet another HiC assembly tool, YARS. And this is what we've done to date with that project. So far, we've generated sequence data from over 7,000 species. This is as of the 13th of May, created 3,404 assemblies. And of those, this has led to around 1,200 publications, genome notes of those assemblies, which are readily available. Now, such project of this size, where we're aiming to sequence everything, as you can as you can imagine, has its issues, has its problems. And one of the big challenges is some of these organisms are very, very small and have limited amounts of material. And so sequencing on any platform is difficult, but with PacBio and PCR-free can be challenging. So we've worked with PacBio for many years, and in 2018 we developed with them a low input protocol, which generally requires around 400 nanograms of starting material, where we size select using spry beads. Of course, for some organisms, we have even less than 400 nanograms. And for a few years, we've used the PacBio's ultra-low approach, which allows you to start typically from around 20 nanograms of input material. However, sometimes we don't even have that. And also with the ultra-low approach, you've got to pay for both the ultra-low library prep as well as the standard library prep. The ultra-low protocol had 2 PCRs, so you had to amplify each sample twice and pool them together, and that was more difficult to keep track of in the lab, and a potential issue if you potentially pooled 2 wrong samples together, which hopefully we didn't do, but it was just challenging in the lab. And after that ultra-low amplification and sequencing, assemblies were quite often fragmented, in that the quality of those assemblies wasn't as good as those from the standard protocol, where you have lots of input. So we sort of internally thought about how we could improve that, and sought to take advantage of some other work that myself and my group were doing in parallel, where we'd done a study with PCR enzymes to evaluate a range of enzymes to see which enzymes are the best enzymes for the PCR step in next generation sequencing, in terms of keeping the fragment size, but also amplifying regardless of DNA content, repeat structure, and GC content. So to do this, we approached every manufacturer. We got them to recommend their best enzyme, and for both short read and here for long read, we used each enzyme with a series of test fragments to assess for both yield and genome evenness of coverage across that genome. For the long read application, we took size selected long fragments, around 12 to 20 kb fragments of yeast DNA, and amplified them with a range of enzymes under their recommended long-range conditions. And the histogram above shows the yield for each enzyme. So this varied, as you could see, quite wildly. After with 1 nanogram of size-selected input DNA, and after 15 cycles of PCR. So as you can see, some enzymes virtually produced no product, whereas the best performer here, Replica, gave the best yield. So armed with those enzymes that gave enough DNA for sequencing, we sequenced those on PacBio, and we looked to see which gave the best insert length after sequencing, and Replica maintained the fragment size the best, followed by Takara's Terra enzyme. And then we assembled that data and looked to see which enzymes gave the most intact and best assemblies. And replica followed by Terra gave the least number of contigs in the best length. And so that was what we published in Microbial Genomics last year. At more or less the same time, we were given the opportunity by the PacBio team to get early access to the newer ULI protocol, which is now Ampli-Fi. And we sought to use that under a range of different conditions. We were convinced that replica was a good enzyme and gave good yields and good fragment length integrity during amplification. So we wanted to test that alongside PacBio's recommended KOD extreme enzyme. And we also wanted to test a range of different inputs, because one of the challenges with the old ULI protocol was that there was just one input. And say if it was 400 nanograms, we'd have used our low input protocol, but we might, for any given organism, just have 200 nanograms. So it seemed kind of silly to take 20 nanograms of that material and do lots of PCR cycles, whereas in theory we could just take all of it, 100 nanograms or 200 nanograms, and just do a very few number of PCR cycles and get minimal bias. So in this experiment, we took this actually summarizes two experiments where we took a subset of our Tree of Life reference genome panel containing oak tree, Quercus rubor, snail, Bioinflaria, the algae, Dunaliella, as well as the two-spotted ladybird, Adalia bipuncta. And we did at the top a trial of different input amounts. So from at the top, 95 nanograms down to 20 nanograms. And we also did a bake-off, if you like, a comparison of KOD versus replica enzyme. So we did that for a number of different species and we sequenced them. We got good yields, good mean HiFi read lengths, and we did assembly and analysis, which Ksenia is now going to talk us through.
Okay, so hi, my name is Ksenia Krasheninnikova. I'm a senior bioinformatician with the Tree of Life assembly team. We have compared different input data sets produced with the Ampli-Fi method, and I'm going to show you the results. We have looked at 10 data sets produced for 4 different species from the Tree of Life R&D panel. The species are an English oak, a freshwater snail, an algae, and a two-spot ladybird. 3 of the species are diploid, and the algae is a haploid genome. The genome sizes vary from 211 megabases for the algae to 850 megabases for the snail. The experiments were performed with Ampli-Fi workflow for the different input mass and using 2 different enzymes, CodExtreme and RepliQ. Before I start, I wanted to go through the plots that I will use in the following slides. These are called the K-mer profiles and are part of our standard QC checks. K-mer is a short subsequence of read with a fixed length. For K-mer profiles, we use K-mers of length 21. The plot on the left is an example of a K-mer profile for the whole read set. We can list all the unique different K-mers present in the read dataset. For each unique K-mer, we can count how many times it is present in the dataset, which will correspond to the K-mer coverage. This is the value of the horizontal axis. If we fix the value of the coverage and count how many different K-mers with this coverage are present in the dataset, we will get K-mer frequency. K-mer frequency corresponds to the value on the vertical axis. In this example, we have a K-mer plot for a diploid genome. It has two peaks, one haploid and one diploid. There is a small area corresponding to the genomic duplications. And there is an area with very low coverage K-mers corresponding to whatever errors and biases are present in the dataset. The plot on the right is a K-mer plot useful to evaluate an assembly. The axis and the shape of the plot are the same as in the previous one. The colors on the plot reflect how many times the corresponding K-mers are present in the produced assembly. In this case, part of the haploid K-mers are colored black, which means they are not present in the assembly of this haplotype. This happens because they were removed from the primary assembly into the alternative haplotype. And now I'm going ahead with showing you some slides on the analysis that we done for the Ampli-Fi data. First, we compared how the different input mass affected assembly quality for the oak and for the snail. On this slide, you can see results for 3 different experiments using 95, 59, and 20 nanograms of input DNA. The dataset with the highest input mass on the left has 15% more elevated yield, but in general, results look very similar. For all the 3 datasets, we performed independent runs of HiFi assembly. The results are fairly similar as well. It's notable that 20 nanogram dataset resulted in a little more fragmented assembly, and I think this is quite typical for what we have seen in the previous results. On this slide, you can see the K-mer plots for the produced assemblies. The top row is just for the one haplotype, and the bottom row is when the assembly of primary and alternate contigs were combined. Ideally, for the primary assembly, we would expect that diploid K-mers are all represented once in the assembly. However, for all of them, we can see the part of the diploid K-mers are missing and part of them are represented twice. This indicates that HiFi was not able to separate 2 haplotypes very well for all the 3 datasets. These are similar results for the snail genome. Read length and yields look similar again, and the smallest dataset didn't sequence worse than the largest one. Here is the produced assemblies for the 3 runs of the snail genome. It's worth noting here how, with the decrease of input mass, the assemblies become more fragmented, and contigan 50 decreases as well. Also, the sizes of the primary assemblies are all a little bit elevated, as we know that the snail genome size is 850 megabases. This is probably because plenty of the second haplotype was retained in the assembly, which is also reflected in the higher Busko duplication score. Indeed, if we have a look at the K-mer plots for the assemblies, we can see that plenty of the diploid K-mers that should have been separated into alternative haplotypes are kept in the primary assembly. But again, the results don't differ among different input mass. Next, the focus was on the comparison of performance of codex-trem and replique polymerases for the ladybird and for the algae. We will start with ladybird. The K-mer profile for replique looks cleaner. See the arrow area and how well the 2 peaks were separated, very similar. Notably, the assembly results for the replique-based data are more contiguous, with N50 4 times higher than in the code dataset. The assembly K-mer plots have small differences, like the amount of duplicated sequence that was supposed to be unique. But on the large scale, the results are quite similar. The algae was a haploid genome, so we get only 1 peak on the read K-mer profile. Replique reads have slightly higher yield and slightly higher read N50. In this case, assembly results demonstrate that for code-based input, there is higher N50, but replique-based one has less contigs. Both K-mer plots for the assemblies look nice and clean here. Finally, we compare the amplifier sequencing with the previous ULI datasets for the ladybird and for the algae. For the ladybird, the old ULI dataset was sequenced from a different sample, and the reference genome assembly was obtained using PacBio low-input sequencing on a third unrelated sample. As an example of Ampli-Fi runs, we used one code and one replicate dataset, but downsample to match the old ULI data. The read and 50s were similar for the new datasets comparing to the old ULI. But the camera plots demonstrate that the replicate results have less bias and peaks are better separated. Based on the read mapping to the reference assembly, we didn't observe any significant dropouts. We also observed that the old ULI dataset has slightly elevated rate of chimeric reads comparing to both new datasets. All three datasets had the same coverage, 22x per haplotype, and the most continuous assembly with the highest N50 was produced from the replicate data. Similar analysis was performed for the algae dataset. In this case, the old ULI data was produced from the same biological sample as a reference assembly. The old ULI data had slightly higher read in 50. Here, for the algae, the slightly higher dropouts were observed in the replicate dataset. It is similar as for the ladybird, and even better observed that the rate of chimeric reads is higher in the old ULI. For these three datasets, the highest quantity 50 was observed in the core data. And this is all that I had to demonstrate today. And the main observation could be that the decrease of input material doesn't largely affect the quality of the data. However, higher input amount helps to produce more contiguous assemblies. In comparison to the previous ULI method, the results look similar or better. Cord and replicate polymerases produce similar results on the two datasets. Thank you for listening to my talk.
All right, hi, I'm Scott Geib. I'm the research leader of the Tropical Pest Genetics and Molecular Biology Research Unit in Hilo, Hawaii, working for USDA-ARS. Today, I'm going to be talking about Ampli-Fi-based low-input, high-C sequencing for de novo assembly of arthropods. So within USDA-ARS, we work a lot on tropical pests of crops and commodities. And as part of what our research unit does, we do a lot of foundational genomic research development. So within that, we do a lot of development of whole genome sequencing of agricultural pests and beneficials. This is largely contained within the Ag 100 Pest Initiative, which is working on insect pests of crops and commodities, trying to make reference quality genome assemblies for the top 100 US arthropods and agricultural pests. We also have the Genome 100, which is a newer initiative which aims to sequence and assemble and annotate the genomes of 100 US bee pollinators. So other than just the European honeybee, there's hundreds of different pollinators out there working to pollinate crops, to perform ecosystem services and things like that. And so essentially these projects are at a foundation focused on generating a reference genome that's at a chromosome scale that people can take from there and do whatever they want, help address agricultural problems. While you can say we're just sequencing a bunch of insects, it's really not just rinse and repeat. Many of our specimens are novel, rare, and may be represented only by a single specimen. That specimen could be a single representation for the species or a strain or a population. And so we're really focused on how we can get as much as we can out of a single individual. And then also how we can repeat this over time, even though all of our specimens can be very different. So in the ag pest context, we can do things as large as like the Asian giant hornet or the desert locust or large grasshoppers and things like that have lots of material to work with and kind of follow that standard HiFi, high-C pipeline that we're kind of used to. But we also have other specimens like psyllids, aphids, mealybugs that are tiny, recalcitrant to sequencing, and really require us developing different paths to success. Despite that, we've sequenced many hundreds of organisms across the US that are important to different cropping systems. A lot of times you think of these things as just sequencing the genome and creating chromosomes and putting an assembly out. And we do that in many senses. So you can see here some examples of where we're really just generating reference genomes for people to take and do something with, or to be part of a larger analysis. But we also often have very specific questions in evolution, systematics, and diagnostics. So developing source determinations, phylogenomic context, resolving species complexes. In functional genomics, so things like understanding insecticidal resistance or mapping specific traits in populations or species. And then also multitrophic interactions. So really thinking of it as a holobiont and looking at microbes that are associated with insects, viruses, beneficials, and things like that. On the genome space, we also kind of fall within that same context, where in some cases we're just creating reference genomes for conservation importance, for larger evolutionary analyses and things like that. But there are also very pointed questions we have as well, understanding how the insects are working in order to be the best at what they do in terms of pollination and ecosystem services. So a lot of this is just feeding our hungry sequencer. This is our Revio system in Hilo. We typically, with the output of a Revio, you can do 4 plant or insect-sized genomes per day. That's kind of conservative without any barcoding on each SMRT Cell. It's about $1,000 per genome if you run 1 SMRT Cell per genome. And then in 2025, we updated to the Spark chemistry. So now we're achieving often above 100 gigs per run, gigabases of data, which has now created, if you throw 1 insect into there, you're often generating way more data than you actually need for that insect. So we're interested in thinking about how we can better leverage the system to generate more data for that individual, especially if it's something that we only have one of or are difficult to obtain, and we have some really good questions about how they're working or want to know more about them. So of course, we usually think of the Ampli-Fi protocol about using less DNA allows you to sequence tiny things. And so we do that at times. So the Ampli-Fi protocol is really just at its core attaching these Illumina-esque style adapters to the genomic DNA. The only difference here is the insert size that we're targeting is much larger than you normally would for an Illumina library. But in doing so, it allows for robust amplification of that library, as well as indexing if you wanted to put more than 1 sample in. And then we modify this by adding a KOD extreme hot start polymerase, which is able to amplify much larger fragments robustly than sort of your typical Illumina library prep. But then you basically turn that library into a SMRT-Bell library and sequence it on PacBio. So we've had some really good successes with this. So we have this tiny parasitoid wasp. It generated much less than 50 nanograms of total DNA. And our Ampli-Fi library, you can see that really doesn't look that different from a HiFi library. We're really able to reach out and achieve long fragments if we go in with high quality DNA. And so our HiFi run yielded over almost 110 gigs of data. And the only thing that I would say that's different here than what you might expect in a typical HiFi run is maybe our fragment length is slightly smaller due to a slight amplification bias for targeting smaller fragments and losing some of the larger ones. But one of the other positive notes of the Ampli-Fi is you're generating brand new happy DNA. This PCR amplified DNA seems to sequence very well on the sequencer and produce these very high outputs. So as we dig into the data, we can see that 99.8% of our reads contain the amplification adapter. So they are, in fact, our Ampli-Fi library and not something else. And by sort of controlling the amplification of the library, we can get a really low PCR duplication rate. So only 2.8% of our library had duplications. So we ended up with over 100 gigs of data for this WASP that we were able to go forward with analysis. So thinking of this in this concept of holistic genomics, or how much can you get from one bug, is kind of what I like to think about. We're really trying to think about how we can get our genomic sequence at a core, but then add on a lot of extras, things like HiC data to give a chromosomal context, microbial associations coming from either unbiased metagenomic sequencing or 16S sequencing, looking at the gene structure and content using things like isoform sequencing or isoseq, and then some epigenetic signatures you can get if you're able to sequence the native DNA. And my question is maybe, if we have to take the whole fly and generate a standard HiFi library, could we instead take in order to get enough DNA to do robust sequencing and get high quality data, it may be that we can take just a little bit of this sample, amplify it, and generate a genomic backbone. But then it opens the door up for us to do some of these other techniques. So the rationale here would be that using less DNA going into the de novo assembly, you might have more material for other things in the sequencing project. So you can kind of cut this thing up, maybe not exactly in half. And so you could do genomic DNA extraction and go SMRT Bell route, but maybe instead you could replace this with an Ampli-Fi, so you're saving more material for other things. You can do RNA sequencing, isoseq. You can do metagenomic profiling with 16S, or just look at metagenomic context to your genomic DNA sample. And then we're really interested in how we can add in this HiC library prep, looking at formaldehyde fixing nucleic prep. And then potentially these all could be pooled together or sequenced in some way on PacBio sequencing. So we're really removing that context that PacBio requires a lot of material going in to get good results off the sequencer. And with the high outputs of the Spark chemistry, we have a lot of extra reads, I guess you could say, or output to deal with, where we're trying to figure out how we can leverage for more information. But traditionally, you can't sequence HiC data on PacBio. So HiC library preparation, traditionally you have these interactions between different parts of the chromosomes in the nucleus, and so we cross-link the DNA to hold those conformations in place. And the traditional HiC, you'd cut it with these restriction site cutters. You then add a fill-in to kind of blunt end the thing and add a biotin tag. You ligate that together, and then you shear it down to create Illumina-sized bits that you can sequence in paired end sequencing. And those two paired ends give information about two different locations in the genome that had an interaction between them. So this, in my mind, is a little bit oversimplified in what's really happening in your sample. In reality, these contacts aren't a one-to-one contact, but a many-to-many. And so here you could see you could create a path between red, going to blue, going to purple, going to green. And all of those interactions are important individually as you would capture them in HiC data, but could become more information rich if we could capture them all in one context. And so that's one of the ideas behind what is trying to be developed for PacBio sequencing within the CiFi sequencing, is to not just only enable HiC data on PacBio, but also add this extra layer of these multiple contacts into the sequencing. So the Dennis lab at UC Davis, Megan and her group kind of developed the CiFi sequencing, which is HiFi based. We could call it HiC sequencing, but it's really more of a 3C library sequencing. The idea here is you do the same chromatin cross-linkage, you digest the chromatin with restriction enzymes, and here you can pick different restriction enzymes than you'd maybe typically pick for HiC, ones that maybe cut less frequently, create larger fragments. Those fragments are then ligated together, but we do not add biotin, so there's no enrichment step using biotin tag to pull down the resulting thing, but then everything else kind of goes along the path. With the exception of after cross-link reversal, instead of shearing this fragment, which contains multiple contacts in it, we just purify that and target that entire fragment with many segments for sequencing. So you can repair that and get it ready for sequencing. This would be what the input for like a POR-C library would be. It would be the native DNA that's undergone cross-linking, digest, reverse religation, reverse cross-linking. And we find that this doesn't always sequence the best because this is DNA that's gone through a lot. It's difficult to remove the cross-linkages completely. We find that the fragments going in don't always generate exactly what's coming out. So using the Ampli-Fi technology, we're able to PCR amplify this product. This enriches for DNA that's intact and of high quality, and then all of the new copies of it become very sequenceable. So we have this fresh, high-quality template DNA going into SMRTbell library preparation and then into PacBio sequencing. And the resulting fragment has all these multiple segments into a single read, so giving information about multiple contexts within the genome. So the Dennis lab, they work on human cell lines mostly, and this is just to show that the HiFi-based sequencing is kind of on par with HiC in context. So you can see coverage across the genome pretty standard. Between the standard HiC and CiFi, you're getting even coverage across the genome. And then as you pick different restriction enzymes, you can either pick to have many segments contained within a read, but have shorter segments, or have fewer segments contained in the read by picking a less common cutter, but having longer segments. It's really your choice of what type of information you want. And the idea being that longer segments may map more uniquely in the genome, but more context may give you more information for your downstream analysis. So a CiFi read here, you can see this is about a 10 kb read. It's broken up into its read segments, and they're mapping uniquely across a large segment of a single chromosome, as we would expect. And then as we do pick larger fragments, we also get improved coverage across the regions that can be difficult to map to, like repetitive elements. So I invite you to look at the preprint that's up on bioRxiv to get some more information about the CiFi in general and some of the applications in human. But I'm going to talk about how we're using it for some low input insect tissue. A lot of this work, the wet lab being done by Renee Corpuz in our lab, and Shenisem doing a lot of the bioinformatic analysis. So we have our fly again, we cut it in half, we formaldehyde fix, and we go into the nuclei prep for CiFi library preparation. So we did this for a variety of insects. We have moths and beetles and flies. And following kind of the standard protocol, we ended up with read length distributions that targeted around 5 kb in length. Still pretty good, getting tons and tons of HiFi reads. And the fragment links being on par or better than what we would maybe expect to see from something like a POR-C library, containing lots of segments per read and allowing you to do a lot of analyses for whatever you want to do with that CiFi data. But we also wanted to try to see if we could reach out and get longer reads. Also, by having a larger library, we could potentially pool it with a HiFi library. So we did a size selection around 9 kb, focusing on this region of the curve or the distribution of read links. And we ended up getting something that looks like this. So we have now, we're targeting more of these 15 or 12 to 15 kb links. We didn't quite cut off all of the smaller stuff. And we go back and resequence that with the 9 kb targets. And you can see we're really pushing our read length up. And we actually increased the number of reads coming out of these libraries as well. I think by removing some of the very, very shortest read lengths, we get slightly more efficient libraries. I'm going to describe the application of this in a system of tropical fruit flies. So we work a lot on these differentiated fruit flies. They're pests worldwide with different genera basically targeting different regions of the world, be it Ceratidis, Anastropha, Bactrocera, or Zugodacus. They're a really big threat not only to directly damaging crops, but impacting the export of crops globally, and often impose quarantine treatments and different things to stop the movement of the crops worldwide. So one of the most foundational ways that they control these globally is using the sterile insect technique, or SIT. And they do that using what are called genetic sexing strains, or GSS. A genetic sexing strain has a translocation between the Y chromosome and the autosome that allows for sex linkage of important traits that can be used to not only to kill, so temperature-sensitive lethal, or also mark the pupae, so the pupae end up being different color, so that we can sex sort for male-only individuals that have a known pupil color and aren't killed by elevating the temperature. So we take these males now that we have male only, we irradiate them to cause sterility, and then we release these by the millions or tens and hundreds of millions. And when they mate with wild females, they produce no offspring, embryos don't develop, and the populations crash. So what we're trying to do here today is look at the nature of this chromosomal breakpoint and translocation using a HiFi CiFi approach. So we took our fly, like I described, we cut it in half. We were able to use a standard HiFi library prep and pair that with CiFi library. And we paired this to have a little bit more, or a lot more, HiFi data than CiFi data at about a 1-to-4 ratio of CiFi to HiFi. We fed this to our hungry sequencer and generated a library with a redistribution looking like this, where you can see the proportion that are coming from the fly being kind of this larger fragment, and then some of these smaller peaks are more indicative of what we're getting for the CiFi library. But we're able to sequence it all in one go, generating about that 4-to-1 ratio of HiFi reads to CiFi reads, with the expected shorter read lengths in the CiFi libraries compared to HiFi. So this read, like I said, contains segments that are all proximity ligated together. And so using a POR-C protocol, it works the same in the HiFi data. We're able to break this apart a bit. We do have to modify it to allow for better mapping of the higher quality HiFi data. And in picking different restriction sites, you can see that we again get either more segments of shorter length or fewer segments of longer length. And you can really choose your own adventure of which of those works best in your situation. It may also depend on how much data you're able to generate. In doing so, we were able to completely phase the Mediterranean fruit fly genome into its 2 haplotype components and capture both a wild type chromosome 5 as well as one containing the translocation with the Y chromosome. We did this by just basically taking the HiFi ASM and feeding it CiFi read pairs as Hi-C data to allow phasing of the data, and use the dual scaf option as well to fully resolve these haplotypes. We then use the CiFi data a second time and create YAS-like interactions, or Hi-C-like interactions that can be fed into YAS. So YAS is yet another Hi-C scaffolder, and this allowed scaffolding individually of the 2 haplotypes being resolved from the assembly. This, like I said, allows us to explore structural variations around the sex-linked translocation. So here you can see, these are now kind of put together where hap1 and hap2 are adjacent to each other. And you see a completely wild-type chromosome 5 as expected. But we additionally see a chromosome 5 with this Y chromosome. And you can see contacts between the Y chromosome that's down here and the 5th autosome, as well as the reciprocal of that as a big, huge piece of the 5th chromosome being put into the Y. So this is really cool that we were able to resolve this and get really good mapping of our CiFi data into the Y chromosome, which normally is difficult to get Hi-C data mapping to because it's highly repetitive. So an example of this, you can see that we're getting better. So this is the same strain. We're able to take Hi-C data and CiFi data, and we're getting better resolution across these regions where we really get strong Hi-C dropout. The CiFi data just kind of chugs along. We also don't need a lot of this data to get information. So as we reduce the number of read pairs, we can see where our genome assembly kind of falls apart. And it's all the way down at 250,000 read pairs to scaffold assemblies that are of the contiguity that we're talking about coming from HiFi. Just another look at this CiFi versus standard HiC mapping to the Y chromosome. So this is the portion of the Y chromosome that has a bit of the autosome on it. So you can see here, this is an interface between Y chromosome and autosome. And the HiC data basically drops out completely. You'd never be able to characterize that as a translocation, whereas the CiFi data just keeps mapping along, and you can see that there's contexts that support this joint. And the reciprocal of this, you're seeing a portion of the Y chromosome stuck into the middle of the fifth autosome, and the same pattern holds where you can kind of see this complete dropout of the HiC data, but the CiFi data continues on. That being said, we do see these bands where we get no coverage, and so we wanted to investigate that further. And so we do see coverage dropouts in some of our CiFi libraries, but this seems to be related to lack of restriction sites in these heterochromatic regions. And so you can see the how many restriction sites we're seeing across the positions here, and a complete dropout that's matching with this banding here. So it's not the nature that the sequencer isn't able to sequence the region. It's more that there's no restriction sites there, so we're not collecting any data. You could modify this by picking a different restriction site cutter to target this region based on genome structure that you already know, or look at some restriction site-free approaches, things like DNase-1 Omni-C type that should be more continuous across the genome. And they do seem to be feasible with CiFi. Data has been generated using these approaches and is currently being tested and analyzed. So the real caveats and takeaways here is CiFi is an application of Ampli-Fi for whole genome amplification-based PacBio sequencing that allows for efficient sequencing of HiC-like libraries on PacBio. These libraries, though, contain many associated proximity ligations, not just a one-to-one, but a one-to-many, allowing for combinatorial associations being made from each read. And the length of each fragment is driven by the restriction enzyme you choose, or maybe the conditions that you're doing something like an Omni-C under, but is typically substantially longer than traditional HiC reads, and so seem to map better and more uniquely across the genome. And in many cases, due to the high output of the Spark chemistry, a CiFi library could be spiked in to a standard whole genome sequencing HiFi run, particularly for us where a lot of our insect genomes are below that 1.5, 1 gigabase size. And so we have extra data and can add some chromatin conformation information to a sample. So with that, I want to thank our lab in Hilo. A lot of this work, again, I said, was coming with help from Ksenia and Renee in our lab, and I'd be happy to take any questions. Thank you.
Everybody, I know we're kind of at the end of the hour, but I think we're willing to stay on for a few more minutes to help answer some questions. So there's been a few to come in the chat, but if you have one, please use the question panel up there in the control panel. So I think I can take a few of the first ones that I've seen come through. So one user asked if they can use pipette sharing method for the Ampli-Fi protocol. We did explore this early on. I think the issue with the pipette sharing method is it tends to create slightly larger fragments than what you can accomplish with a G-tube or megaruptor. So that way, it's going to kind of bias the PCR step because the enzymes are not going to be able to sufficiently amplify those longer fragments. So what ends up happening in that type of sample, you just amplify some of the shorter fragments at the expense of the larger ones. So to get a more cleaner distribution, not a bimodal distribution, it's better to use the G-tube or the megaruptor systems for that. Let me see a few more questions coming in. Okay, so we had a couple of questions on FFPE material. So do we recommend sharing an FFPE? Usually not. FFPE samples are usually fairly degraded, very fragmented. So most of the time, you can just simply skip any kind of extra sharing step for that. Okay, so also with FFPE, do you recommend implementing DNA damage or repair step prior to end repair and ATLing? Yeah, so we would repair those prior to amplification. It does tend to help that process.
let me see here. So Scott, do you find, is there like an optimal restriction enzyme for doing the scaffolding? Like, I mean, do you prefer the longer segments or more contacts to kind of help? Because the longer segments would give you sort of improved mapping, right?
Yeah, so that's something we're definitely still exploring. I think a lot of it comes into what question you're trying to ask. So in the example I gave, I think the longer fragments are kind of helpful for the translocation, specifically in the Y chromosome, because it's very hard to map high-C reads in the Y chromosome. But if you're just going for volume of data, you could pick a smaller fragment, which is going to get you more context for the same amount of sequencing being done. So you could also potentially prepare two libraries or something like that and get a best of both worlds kind of situation.
Okay, great. And I think there's, related to that, there was another question that came in about, interesting approach, though your point about restriction of site dropouts is pretty important. So as you mentioned, is DNase-1, could you use DNase-1 as an alternative? Is that being tested? Or like, a tag mutation approach like TN5, have those been examined?
Yeah, so for sure, like an OmniC style approach, Megan's group has generated some human data on that, and we're in the process of generating similar in some of our insects, and that could be a nice alternative to restriction site-based approaches. One of the things there is you just got to kind of dial it in to not just generate really short fragments. We kind of want to retain those longer fragments, and it might be a little bit of work, across a broad breadth of specimens to get kind of the same result across everything.
Yeah. And so, for everybody else, stay tuned. We're going to do more work on evaluating, taking more of a DNase-1 or OmniC approach on that. So another question came in, are the protocols working as well in the Vega system? I can take that one. Yes, the Vega system, works just as well as the Revio system. It's just going to be a little lower throughput, but for many of these, tinier organisms, like for example, what Scott was showing, the Vega system would give you, more than enough output for that. Scott, I think, another question for you. Amazing talk. Always enjoy to learn about your work on insect genomics. What is the oldest sample you've successfully sequenced with Ampli-Fi? Have you had much success with non-freshly collected frozen material, aka maybe like old pin museum samples, things like that?
Yeah, we haven't really done any museomics kind of stuff on these kinds of samples, but we certainly could try. We do some work on insects that are like trapped as part of surveys for invasive species, but really haven't thought about applying some of the PacBio sequencing to it, but certainly could give it a go. I would guess we'd probably not be able to achieve beyond, you can't make the DNA bigger. If it's already sheared going in, it's never going to get any bigger. So you'd probably top out around maybe 3 to 5 kb, but it would all be dependent on the sample going in and stuff like that.
Okay. Yeah, so there's another question came in. Can we push the genome size? That would ideally be assembled using this low input system. Yeah, sort of. So like what I try to present in my section was that, with this new protocol, if you go in and you follow the protocol, you can generate, several, 1,000 nanograms of amplified material. At the end, that should give you enough to run multiple smart cells, especially if you're using the Revio Spark chemistry. And that will provide enough yield to then do some of these larger genomes up to maybe even 4 gigs and stuff like that. If you're going really beyond that, you can maybe do 2 perhaps in parallel with whatever your starting material is. But usually these long, if it's a large genome, it's usually polyploid, so you probably want as long, longer fragments than probably what you can achieve with amplification to really fully resolve some of that complexity there. Let me see what else. Okay. Another question about, okay, so from the presentation of Mike Quail's group, RepliQ showed similar performance to KOD Extreme. Would that be including the PacBio protocol as an alternative? Yeah, so the one way we kind of approached this protocol was that we kind of knew going in that, one enzyme may not be the best for every possible organism there is in the world. And so we've kind of retained a little bit of flexibility to allow users to kind of, interchange enzymes depending on maybe the system they're working in, right? And so that's why it was really valuable to see, the group at Sanger, their sort of presentation, because they kind of went in depth with like exploring a lot of different enzymes, right? So as you saw, like RepliQ showed, slightly better performance on some organisms, whereas KOD showed the opposite, right? Where it was better in some than RepliQ. So we feel like we went with KOD Extreme because we felt like this is the best overall enzyme. If you need to pick one to just go with to apply to everything, we especially saw that like in the human case, it sort of outperformed anything that we tested. But there might be, small differences with certain organisms, so it might be valuable to be able to interchange it, or if you have any problem accessing one enzyme over the other, depending on what market or what region in the world you are in. Okay, I think we have time for maybe one more question, then I think we will need to call it in here. So let me see if anything else has come in. So maybe this one's for you, Scott. DNA extraction from insects can be a real challenge. Are there lessons learned from your experience that can be shared with the community?
Yeah, so we do a lot. I mean, beyond this, just generally, we do a lot of DNA extractions. We try to just generally make the extraction process itself pretty consistent. It may be picking certain tissues of the insect to do. A lot of times less is more. So you might be tempted to throw the whole bug in and do a whole extraction, but there can be a lot of weird compounds and inhibitors and things like that in different parts of the insect. We find in like Hymenoptera that there's some eye pigments that seem to mess with things. So we may just go in and dissect out a little bit of thoracic material and do the whole library from that. And having less really good DNA is probably better than having a whole lot of DNA that's got some problems with it. Really focusing on how clean and pure the DNA is as well as going in, because you are sequencing those exact molecules, is really critical. If it doesn't look good, do some steps to clean it up before you proceed, because it's kind of garbage in, garbage out can be with some of the sequencing.
Yeah, definitely. So like, I think as you mentioned earlier, like the Ampli-Fi protocol is great, but it's not a cure for like, if you're already starting with really highly degraded DNA, it can't make that DNA larger, right? So yeah, a lot of focus on getting, good quality DNA out, at the beginning, and being able to control that shearing process, will definitely improve any kind of assemblies, therefore like downstream by increasing that average read length. Because most issues people run into is just like, it's not necessarily, the protocol itself. It's just like if you have a lot of short reads, you're just going to have a very, you're not going to have a continuous genome assembly, right? The contiguity is going to really suffer, and you're going to have a lot of contigs. All right, I think we're kind of. Oh, sorry, go ahead. Yeah.
I was just going to say, we have selected some of these libraries on the blue pippin as well to kind of remove that, which isn't a part of the protocol, but if you see it there, it can be something that can.
Yeah, it can certainly be helpful. Yeah, it's a good point to make. Like, yeah, obviously we if you're starting with good quality material, the bead based cleanup is pretty much all you need. But yeah, for certain samples, the gel size selection step will definitely help push those read lengths up. And that's another good thing about the protocol generating all that amplified material, is that usually you'll have enough to actually even run through gel size selection. And then if you're using like the Revio Spark chemistry, you need very little of that to actually load onto the instrument. So that's another definitely option out there. Okay, I think with that, we're definitely at time. So I want to thank again everybody for attending today's webinar. A recording of this will be provided to you, so just be sure to check your inboxes in the coming weeks for that link. And for those questions we didn't get to, I'll try to follow up on those. All right, thank you again, and everybody have a good day.